April 13th, 2020, week 12, just gotten back from our so-called spring break. We're still sheltering in place with the coronavirus pandemic. So this is the second virus PowerPoint for this week. The first one was on the immune response to viruses, kind of focusing on the pandemic coronavirus, COVID-19. This one is just a real quick one on some of the diagnostic tests that are used to detect either viral infections or antibodies to viruses, right? And then the third one will be on just a real brief overview of viral vaccines. So folks, what I would suggest is that you either print out or look at the study guides that go with each of these PowerPoints, because you'll see what you're asked to know from the PowerPoints. It's much less detailed than the PowerPoint slides themselves. I tried to, put some links into the PowerPoint, so those of you that maybe are interested in some of these techniques, you'll have some good links to look at. But we just want to know the basics for lecture exam 3. So what I'll try to do, folks, is look at my study guide for the diagnostic test for viruses, and then we'll try to find the slides that will help us answer them. So the first question, you guys, is really the only question that deals with the nucleic acid-based tests. So these tests are PCR, reverse transcription PCR, and real-time reverse transcription PCR. This is the test they're using, or was one of the initial tests they were using to detect the novel coronavirus, COVID-19. And I was able to just peek at the CDC instruction manual for this that the CDC has posted online, and it's like 50 pages. So it helped me to understand why it's taking so long to get some of these test results back, right? So hopefully they'll be able to develop some more rapid tests. And indeed, you guys, maybe some of those rapid tests will be antibody-based tests. All right, so you guys, the first question is, what do the following mean? Describe each process. So reverse transcription, PCR, and real-time PCR. So folks, this was just from the Canvas announcement that I just kind of went through very superficially, the 3 steps in that real-time reverse transcription PCR that they were using and are still using to try to diagnose people that may be infected with the COVID coronavirus. So folks, we'll just take this one step at a time. So reverse transcription, you guys might recall, this is a process, say, HIV does, but we're going to do it in a test tube. So we're going to take our nasal swab, and if the nasal swab has the coronavirus present, remember coronaviruses, they're RNA viruses, right? So we're going to have viral RNA as our template, and then the reverse transcriptase will use that template to make complementary DNA. So the little c means complementary, right? So that's the first step. Now the reason we have to do that is we want to amplify the coronavirus genetic information. Amplify means to make many copies. And the way we're going to do this is with this amazing process called PCR, which stands for polymerase chain reaction. And in PCR, we're going to take target DNA, and remember, you guys, the cDNA now would encode information for the COVID coronavirus. We're going to use this cool heat-resistant enzyme. One of them is called Taq polymerase. It's a heat-resistant DNA polymerase isolated from hypothermophiles growing in near the hot water springs, the hot geysers of Yellowstone. And we're going to use that DNA polymerase to make multiple copies. Sorry, my little mouse keeps fading on me. Multiple copies of that target DNA sequence that encodes information for the coronavirus. Okay, so this is the polymerase chain reaction. And then the third step of this, you guys, is so cool. This real-time PCR, they have a fluorescent reporter molecule that will fluoresce each time a DNA copy is made using PCR. Will have a fluorescent signal released. So as more and more copies are made, we'll have more and more fluorescence released. And this fluorescence can be detected by a sensor. So even as the process is going on, you're getting a report coming in, okay, this sample is positive for coronavirus genetic information. So lots of fluorescent means you've got, in the original sample, that original nasal sample, the interpretation is there was a lot of coronavirus RNA present, right? So you would say the test is positive, that patient is positive for the COVID. So I think, folks, in these the next few slides are just showing you some of the steps. So we don't have a slide for the reverse transcription because I think you all understand that pretty straightforward. This is just a cartoon of polymerase chain reaction. So this would be our target, our complementary coronavirus DNA made by reverse transcription, and then the Taq polymerase is going to make copies, multiple copies with each round, each cycle. We get a doubling of the copies, right? So we're going to amplify the DNA encoding the coronavirus genetic information, and then so we can get a report of that if that coronavirus information is present. We're going to use this real-time PCR process, and this is so cool, you guys. So hopefully I won't make this too confusing. So in PCR, you have to have two primers that are complementary to your target DNA. So let's say, you guys, this is the coronavirus complementary DNA in here. So we'd have two primers that would anneal. And this is a cool thing. So in addition, we have a probe that's going to anneal to that target DNA. And this probe, on one end, it has a reporter molecule. This is going to fluoresce. And on the opposite end, it has a quencher molecule. So as long as the quencher is near the fluorescent molecule, the quencher absorbs all the fluorescence. So basically there's no fluorescence here. We add our Taq polymerase, our DNA polymerase, and as it's going along using the primers and then making copies of the target DNA, when it hits that probe, the DNA polymerase has what we call an exonuclease activity. It's going to cut out the end of the probe carrying the fluorescent molecule. Okay, and so when that fluorescent molecule is released, right, now it is going to fluoresce. There's no quencher molecule there to absorb the fluorescence. So this fluorescence is what will be detected in the machine in which we're running this cool process, right? So in real time, right, you'll know as soon as you have a PCR going on that is amplifying the coronavirus genetic information, you'll detect it by this fluorescence. So the more fluorescence there is, the more, that means the more coronavirus complementary DNA is present, right? So you would say that's a positive coronavirus test. That patient is infected with coronavirus. So that's kind of amazing. These are just some tutorials, folks. This is only if you're interested in these processes. If you're not interested, don't worry about these. But again, I just tried to provide some links for those of you that might be interested in these nucleic acid diagnostic techniques. So folks, this is study guide question 2. What does ELISA mean? And so ELISA, this is a test that doesn't use it doesn't use nucleic acid. It's going to use antibodies. And ELISA stands for enzyme-linked immunosorbent assays. And basically, we're going to have antibodies linked to an enzyme. And we're going to see in this test that if we add a substrate that's colorless, if it interacts with the enzyme that's linked to the antibody, the substrate will turn a color, and we can detect that color. It might be color, it might be fluorescence, right? So there's different types of ELISAs. There's direct ELISAs and indirect or sandwich ELISAs. What I'd like you to know is, what can these tests answer? What can they tell us? So first of all, you guys, they could tell us, we could set up an ELISA in which in this particular ELISA, we could answer the question, is a patient infected with coronavirus, right? So we'd set up an ELISA, we'd take the nasal swab from our patient, right? And then we'd use the material from the nasal swab in our ELISA to determine if viral proteins or the coronavirus proteins are present. And it would be hopefully very specific, right? And then another way we can use the ELISAs is to determine if a person has antibodies against the coronavirus, right? So now this wouldn't tell us whether the person is still infected or not, but it was tell us whether a person has antibodies, right? So it might be a person was infected, right, and they recovered, they're no longer infected, but the antibodies is kind of the smoking gun. The antibodies is the signal that tells us this person was at one time infected with the coronavirus. Now again, folks, if a person has antibodies, you'd have to run another test to see if they're still infected or not. The reason this is going to be really powerful, folks, is twofold. So let's say a person has recovered from coronavirus, they're no longer infected, they have all these circulating protective antibodies. You might remember in the last video we talked about neutralizing antibodies and opsonic antibodies, right? So one thing we could do is ask that person that's recovered to donate plasma, and plasma is a liquid portion of blood. It's going to have the protective antibodies in it. And then we could transfer that plasma to a person that's infected with coronavirus right now, maybe a person at high risk, right? And so those antibodies then could help protect the person that's currently infected with the coronavirus, right? So that would be a form of immunotherapy. And this is something that they used in the horrible Ebola outbreaks. They found that antibodies from people that have recovered played a really important role in helping to save people that were newly infected. So this could really help us protect some of our high risk patients who are infected with coronavirus. The other thing that I just hope so deeply that we'll get these antibody tests available, and hopefully they can be done cheaply, is another reason is um, if you are not sure if you were infected with coronavirus, especially young folks, right? So often young folks are infected and they don't know, or they think it's a cold, right? Many of us in January and February, a lot of us in our department and staff and faculty and students we were having respiratory infections of some kind. And we've all questioned, it's like, well, could some of us have had coronavirus early and we just didn't know it, right? So if we had a quick, easy, inexpensive antibody test, we could screen ourselves. Those of us that are antibody positive and are well, it would suggest that we have some protection against coronavirus. So then we could go volunteer, like in the hospitals. I feel so badly that I'm not helping, I'm here at home. But if I knew that I was antibody positive and knew I wasn't actively infected, I would love to go volunteer in the hospitals. Not that I could take care of patients, but I could clean bathrooms and I could clean patients' rooms and ERs, just help in any way possible. But I wouldn't want to go if I thought I could get sick and then I become a burden for everybody else. But if I had circulating antibodies, then I could go volunteer my help. Okay, so that's kind of that's me going off on a side path. So let's get back to ELISAs, you guys. So this is called Direct ELISA. So the links, you guys, and like the YouTube videos, they're better explaining this than I am. But let me see if I can do my job. Okay, so in these ELISA tests, often you use what are called these little microtiter plates that have 96 little plastic wells. And so in direct ELISA, what we could do if we had, say, grown the novel coronavirus, the COVID, somewhere maybe in tissue culture, because I mean, they according to our resources, they can grow it in tissue culture. You can harvest the coronavirus, maybe harvest the coronavirus proteins. These are the antigens our immune system recognizes as foreign, and these antigens are what will make antibodies against. And then you would stick the coronavirus proteins, antigens, to the walls of the wells. And then, folks, this is a wild thing. This would be the antibodies from your patient. Now, this would be a little bit trickier because in ELISAs, what you have to do is take the antibodies and then link them to an enzyme, right? So conjugated to an enzyme. So enzyme linked, right? Enzyme linked, right? So then you would add your patient's antibodies to the microtiter wells. And if there's patient antibodies against coronavirus, the antibodies will bind. And then you'd wash really well to get rid of any excess antibodies. And then what you're going to do, this is a cool thing, is you add a substrate. And the substrate is going to start out either being colorless, right? And when the substrate is acted upon by the enzyme changes the substrate into a colored molecule. So you can actually, by looking at the color of the solutions, the microtiter wells, you can tell which patient had antibodies against the coronavirus. Or this could be a substrate that's not fluorescent until the enzyme acts upon it and then it fluoresces. So then you have these microtiter machines that can read either the color or they can read the fluorescent. And that's how you could identify which of your patients have antibodies against the coronavirus. This is indirect ELISA, or sometimes called sandwich ELISA, and this, I think, you guys, is probably what they'd be using if they're looking, if they are developing a quick test for coronavirus antibodies. So again, we start out microtiter plate. Here's our coronavirus antigens, proteins here. We add our patient's serum with the antibodies. The antibodies, if the patient has antibodies, the antibodies bind to the coronavirus antigens. We wash, that's really important. And this is a step, you guys, that actually makes this a little bit easier. Then you would add a second antibody, an antibody against human antibodies. For example, we could raise antibodies against human antibodies in a goat. And then these goat anti-human antibodies are the ones that would be linked with the enzyme, conjugated with the enzyme. Excuse me. So we would then add our second antibodies, the goat anti-human antibodies. So if there's any human antibodies there, the goat antibodies bind. We wash really well to get rid of the excess secondary antibody, and then we add our substrate, right? So again, if the enzyme linked goat antibody is present, linked to the human antibody, right, then our colorless substrate will turn color, or our non-fluorescent substrate would fluoresce, and again, we'd have a machine, or just sometimes by eye, we can detect that presence. So again, this would be an example of a patient that has antibodies to the coronavirus. Now, another possibility, you guys, that's not here, but if you kind of a mind experiment, think of another type of ELISA where instead of having the coronavirus protein stuck to the well, what if we had antibodies against coronavirus stuck to the well, right, sticking out here, right? And then what if we added the nasal swab, the nasal secretions, right? And if there was any coronavirus present, right, the coronavirus proteins antigen would stick to those antibodies, right? And then we might have a second antibody against coronavirus conjugated, right, linked to an enzyme, and we add that conjugated antibody. It would bind to the coronavirus that's bound to the antibody stuck to the cell wall. At our substrate, we get a signal. I know that was a little bit more complicated, you guys, and I'm pretty sure some of the links would actually give that as an example. Okay, and again, folks, this is just so much extra information. But again, those of you that might be interested, also there's a short little section, you guys, in our OpenStack microbiology book in chapter 20. It's section 20.4, and they actually have figure 20.22. They have a little cartoon of an ELISA. Okay, and folks, this next part is going to be bonus. So if you're just feeling overwhelmed, don't worry about it. But I just thought this would be really good for us to mention, because you're going to hear about all these different types of coronavirus tests that are out there. And so the question is, well, how do we compare these coronavirus tests? What makes a good diagnostic test? What makes a poor diagnostic test? So one way we can compare diagnostic tests is to look at the tests, what we call sensitivity and specificity of the diagnostic test. So you guys, in this particular example, um, this would be like an experiment we'd have to run ahead of time. So let me see if I can set this up here. So let's say that we have 100 people we know are infected with coronavirus. So these are our coronavirus infected people, right? Right, going to be in this column. And let's say we have 100 people we know for sure are not infected with coronavirus. So these are going to be our uninfected people right here, okay? And then what we do is we run our specific diagnostic tests on both the group of infected and uninfected people, right? So in this row, in this row, we're going to see who ends up with a positive diagnostic test, right? Okay, so here, you guys, this would be considered a true positive diagnostic test. The test is saying the person is infected and the person truly is infected. But over here, you guys, this is, this is a false result, right? Because the test is saying the person is positive when they're not infected. So that's a false positive. And then down here, we want to look at negative test results. So in this box right here, we have a person that's infected, but the test says they're not infected. So this is a false negative. This is not good, you guys. And then over here, the diagnostic test is saying the person is negative. It's a negative test, and that's a true negative because the person truly isn't infected. Okay, so we can use these values. We'd run this experiment on every different diagnostic test, right? And we're going to get some values. And again, you guys, this is bonus. This is totally bonus questions, okay? So to calculate the sensitivity is going to be the true test positives over the total infected, and specificity is going to be the true test negatives over the total uninfected. Okay, and this is an example, folks. This is, again, it's optional. This is a bonus worksheet. So let's say this is the optional bonus worksheet in the week 12 lecture module. So you guys, let's say somebody is trying to sell you a new pandemic coronavirus diagnostic kit, and you want to know sensitivity and specificity. So this was the results of the lab work that was done. So the company took 100 known infected people and 100 known not infected people, right? And they ran their diagnostic tests. So their test of the 100 infected people, the test identified 80 is truly positive, right? But over here, though, look at this. The test identified 25 of the uninfected people as positive, right? So these are false positive. This is true positive. That's what we want. This is false positive. We don't want that. And then with that same diagnostic test, of the 100 infected people, 20 of them came up as false negatives. And to me, you guys, with this coronavirus, that is horrible. That's not good at all. And then of the 100 uninfected people, 75 came out with true negative tests, right? So how do we get these values, you guys? So what we're going to do to get sensitivity, we're going to take 80 true positives and divide by 100 infected. So that's going to be 80% sensitivity, right? Troubling, because that means 20% of the people who are truly infected think they won't be infected. They'll keep working. Maybe they're working in the hospital. They could go and infect other people, right? So 80 over 100 is 80% sensitivity. And then with specificity, we're going to take our 75 true test negatives, right, and divide it by 100 of the uninfected. So we're going to have a 75% specificity. So that means out of every 100 people that aren't infected, 25 are going to get false positive results, right? So they might have to go into quarantine. Maybe they have to be removed from their family and friends, can't go to work, right? So that's also not that great. So I think the next slide, you guys, is just walking through this. Okay, so remember, you guys, the sensitivity of the test is going to be the 80 true positives divided by 100 infected, so 80% there. Why would you be concerned if this was the only test you gave your patients? Because remember, you guys, that is not good, because 20% of your infected patients will think they aren't infected, right? So they they might not get help at the hospital. They could continue to spread the infection to family, friends, and what if they're medical workers, right? They could spread the infection to people in the hospital. And then the specificity, guys, is going to be true negatives over total uninfected, so that's going to be 75%. So the concern here is that 25% of the uninfected will get false positive results. And again, that might mean they can't go to work. It might cause emotional distress. They can't go to work. They might have to be isolated from family and friends. So that also is not good. But this is the one that really troubles me, you guys, here. Those false negative results. So, excuse me. Okay, so then this last one is a different immunochromatographic test for pandemic coronavirus. Has a 50% sensitivity. So, oh my gosh, you guys, that only means it's identifying half of the folks that are infected, and 99% specificity. So that means 99 out of every 100, 99 out of 100 uninfected people, the test will identify 99 as truly being uninfected. So that means there'd only be 1% that would suffer from a false positive, right? But again, you guys, this would be horrible, because 50% sensitivity, half your infected patients are going to think they're not infected. And again, that means they might continue to go around, spread the infection. If they work in a hospital, they could be spreading the infection to patients. And furthermore, you guys, maybe the person think, oh, my cough is just from a cold, and they don't realize how serious it is, and they might not seek medical help. So that's a horrible diagnostic test. And I think the next slide, you guys, is just the key. Okay, I think that does it, folks. So we'll get this posted, and then the last audio we'll do will be on the third and final viral PowerPoint for Unit 7, and that's going to be an overview of viral vaccines. Again, you guys, I'm going to encourage you to print the study guide first, because the viral vaccine, it has so much information in there, but you just want to know some basics, and we'll go over those basics in the follow-up audio. Okay, you guys, let's see if we can get this posted.